structure, three-dimensional structure of a class of proteins that belongs to this kind of sequence is known. Then only he will be able to tell you. That is why I said, which is 60 to 70% correct. If you don't know the structure of a protein, now if you don't know, meaning there is a database of protein structures, and I tell you till date about 30,000. Now the estimate is so the estimate says there are about 20 to 30,000, actually 20,000 genes have been identified altogether. So 20,000 genes. If there are 20,000 genes, then there are obviously 20,000 proteins. And there, so till date, about 30,000 protein structure is known. Now what I am telling is, how about those whose structure is not known? Then you don't know. That's the answer. Okay? So now I just show you a few of them. This I have already told you that this is a condensation reaction. So it's basically one water is getting eliminated from two such. See, one OH, another H. We remove that, you form a peptide bond. Okay? And then this, you keep on repeating that, so you get a polypeptide, which I have already told you. Now I show you simply, take the pictures. Don't try to read the slides. So proteins, what I was already discussing, gives a sequence information. Oh, this is called as primary structure. Lysine, 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 leucine, valine, alanine, histidine. And depending on what are those amino acids, it is found in nature. Now, how it is found, this I must tell you. These are found through mostly, if not more than 90% cases, it is found through X-ray diffraction experiments. So this is something on which I can immediately tell you probably tens of Nobel Prize. People have got simply by solving structure of a protein. And the latest one I'll show you, where you probably know that an Indian scientist also got a Nobel Prize few years back. His name is Venki Ramakrishnan. Have you heard of Venki Ramakrishnan? Right? So he has also solved a very complicated structure of a protein and RNA complex. Very big. In the real term of the word, machine you can call it. Those are really machines. So, इसमें देखो, so 4 तरीके का structure could be possible, primary sequence. Secondary structure ऐसे होता है, कैसा ये सीढ़ी का माफी, helices, okay? And वो helix का भी एक interesting kind of handedness होता है. These are all left-handed. Alpha helix इसको बोलते हैं. And then you have something called a tertiary structure. This you need not bother much. Quaternary structure also you need not bother much, because at this stage probably I must tell you that whatever protein I have told you right now is, I have told you about one chain. But क्या होता है? Actually you can have, this is probably a good analogy of that. What you could have is two such chains held together as a dimer. And in this case, when you have two such, I haven't yet come to the point how are they held together. We'll come. But only thing I am telling here at this point is they are held together through weak forces. It's not covalent. Okay? So there are very weak forces, but multiple of them. Lot of weak forces makes this adapt very strong. So these two, then I call this protein as a dimer. And since these two glasses and even the water levels are almost identical, I call them monodimer. Clear? Logically? Now there are complexities. I have another protein which I will name, and you know, I know the name of this protein. Those proteins are tetramer. This one, look. But anyway, there are these things, okay? Okay? So, okay, if I make them stand like this. So now this becomes a tetramer. Because 1, 2, 3, 4. And if I call this as alpha, then this is also alpha. If this is beta, then this is also beta. Right? So then this protein becomes alpha 2, beta 2. And the famous protein which binds oxygen and carbon dioxide in our body, or hemoglobin, is exactly like this. It is a alpha 2, beta 2 kind of tetrameric protein. There is an alpha chain, there is a beta chain. 2 of alpha chains, 2 of beta chains makes it alpha 2, beta 2 and forms one intact hemoglobin. And for these kind of proteins, this quaternary structure is important. How? Very obvious, you know. How are they packed? How are they packed? Are they packed like this? Or they are packed like this? Or one is top of one? So there could be many, huh? But this has only primary sequence, which is the primary structure, and the secondary structure, which is how it, whether it has alpha helix, beta sheet, so on and so forth. So this is what all about the structure of proteins, okay? And this I have already told you, this is another picture of a primary sequence of the protein. Take it as an insulin. So here probably that time I forgot that there are ways I can make insulin not in the laboratory but inside a bacteria. Okay, because let's say if I take from like I tell you, liver, any animal liver is a very good source of insulin. But the way you make insulin or isolate insulin from liver is very cumbersome and it's not easy at all. So उसमें क्या होता है? So if I want to make lots of insulin, we need, because insulin is a drug. हमको लेना चाहिए, तो हमको तो मिलीग्राम, ग्राम क्वांटिटी ऑफ इंसुलिन बनाना चाहिए. कैसे बनाएगा? क्या करना है? You can make insulin inside a bacteria. And bacteria can be very easily grown. उसको जाइली डालो, एक फ्लास्क में डालो, थोड़ा खाना दे दो उसको, it will grow. And it will grow very fast. So उसमें से निकालो इंसुलिन. ये चीप भी होता है. These are two gentlemen, you must know at least the name of one, and that is Linus Pauling. Probably you know also. Linus Pauling is a person who is actually pioneer. Both Robert Curry was a crystallographer. X-ray diffraction से वो बनाया. ये आदमी पहला अल्फा हेलिक्स दिखाया प्रोटीन में, हाँ? So I must have done. So, and here is just one example I show you. Why is primary sequence so important? Because if you have one of the amino acids changed from glutamic acid to valine, then you carry a disease called very severe disease. हमारा कंट्री में बहुत सारे लोग suffers from इसका नाम है सिकल सेल एनीमिया और सिकल सेल डिजीज। एंड ये जो सिकल सेल डिजीज है ना, देखो ये सिकल सेल का ये जो आरबीसी है, हमारा जो रेड ब्लड सेल्स है, so red blood cells actually looks like this panel, you know, this biconcave. And you see, well, please find out that this is only one change in the entire primary sequence, and you have a disease like this. This is the secondary structure I showed you. There are something called, you know, alpha helix and all. So, only thing probably I must mention at this point is these sequences or these structures, how are they held together? They're held together mostly by one kind of bonding called hydrogen bond. And hydrogen bond is very weak kind of a bonding. Typically, its energy is between 2 to 5 kilocalories. 2 to 5. Very weak. Okay. But this is the main force that holds together such a big, massive structures of, you know, polymers. Tertiary structure I have already told you. Quaternary structure also I gave you some kind of a example through those glass and all. And these are all those 4 levels of structures that I have been telling you. This is something I have already discussed, but let me just tell you just in one word that when I was showing you on blackboard, on this whiteboard, that when a polypeptide chain is just synthesized, it has not yet folded, then this protein, this state of protein is called a denatured protein. It cannot function. Moment it folds in the correct way, and I have also told you what is correct and what is not, then this protein is called the functional protein or native protein. Okay? So क्या होता है? This one thing I have already told you that this mostly these protein structures are held together through hydrogen bonding, right? So, and this is only 2 to 5. That's the energy. So if you heat simply, these bonds are broken. So that is why if you heat a protein, the protein is denatured. And here probably I give you a, okay, this is something just to show you this I have been telling you that, you know, you can take, I gave you a bottle example, I gave you those 2 glasses and this posters, and that is shape matters, shape of macromolecules. Okay, because these are big molecules and that is why they are called macromolecules. And I skip them and I show you a few structures. Now how these structures are known, like that I will show you. I have already told you that these are all obtained through X-ray diffraction technique. They're all, you have to once crystallize the protein and then take diffraction pattern, and then you have to solve few equations for solve the phase problem, and then got into a structure of 3-dimensional structures. Okay, and the structures look like this. And here I show you the numbers that I have already discussed with you. See, this is a very typical alpha helix. This is not cartoon diagram. This is exactly experimentally solved structure. This is how exactly a protein looks like in this protein. So this is how a protein structure will look like. Okay. Now since time is not in my favor, okay, maybe or studying this protein which looks like this, a cylinder, is easier than studying the structure of this. Don't I make a sense? This is much more complex, right? Because there are odd shapes, there are assembly, there is that fourth level of structural organization. So that makes my life difficult. There is another thing also here in terms of hemoglobin, which I haven't told you, that all of these polypeptide units, they also have one more